PRRS current events session, and we'll go ahead. I think for all of us out in practice, we know that we've just come through the summer months, and now we're headed into the scary time of the year. But our first talk is going to be from Igor Paploski from the University of Minnesota. He's currently assistant professor at the University of Minnesota, his postdoc with Dr. Vanderwaal's lab, and also Mark Wagner, who's a practitioner from the Fairmont Veterinary Clinic and gets to see PRRS every day. They're going to talk to us about the impact of booster immunity pre-farrowing on antibodies and PRRS dynamics in nursery pigs.
So thank you everyone for being here. The session after lunch is a little bit of a challenging one because everyone is a little bit sleepy. I'll do my best to not try to increase your sleepiness. So let me jump back to it. As Dr. Paul mentioned, my name is Igor Paploski. I'm an assistant professor at the University of Minnesota. I'm going to be presenting a work today that we in the university did, but it's a strong partnership that we have had with Dr. Mark Wagner, which is sitting somewhere in the audience and I cannot see very well. There he is. So yeah, the title of the presentation is Impacts of Boosting Immunity Pre-farrowing on Antibodies and PRRS Dynamics in Nursery Pigs. Before I begin with the presentation itself, let me quickly go through the team and the financial support. So over here we have a lot of people mentioned. There are two people that I'd like to highlight. Kimberly Vanderwaal, which is sitting over there, and also Mark Wagner. Again, those are two people that were key into obtaining the animal, the data, the funds for this project. I would also like to highlight the support that we received from SHIC over the year, supporting different people that helped throughout the execution of not only this project, but also others. This project is also a product of funds that we received from the National Science Foundation and from the USDA NIFA. PRRS is challenging to control. I won't preach to the core here, right? You all know that, but why is it that so? So this is a map of the swine density in the US. And as you can see, a lot of the swines are grown in a very restricted geographical area. This is associated to where grain is grown. So feeding these animals, it gets cheaper if you're raising these animals on these regions. But what ends up happening is that these regions have very, very high swine density. Paired to that, the way that the American swine industry has organized itself is in a manner such that animal movements are required. The industry does not work without moving animals. And this interconnectedness between the regions, which is shown over here in the bottom, allows for pathogens that are present in one region to move to another, simply because of the fact of how we chose to structure the industry. Another challenge for PRRS control is the genetic and antigenic diversity. So this plot over here on the right shows lineages and sublineages of PRRS across time, and then the different colors are these different lineages and sublineages. And as you can see, these colors change over time. My point here is that there's never a single, or there's not a single PRRS virus that has been constant throughout time. There's always been changes on those. And that brings a challenge for us because, well, the PRRS that we're talking about is always a little bit different. Another example, another slide trying to illustrate this point a little bit. Genetic and antigenic variability of PRRS. PRRS is one of the viruses that have the highest evolutionary rates among the RNA viruses. So those are many different RNA viruses, and I highlighted over here PRRS. It's up there. This does bring repercussions to us because this is how, or one of the mechanisms by which these different lineages and sublineages of PRRS emerge. But this also brings other complications, which is antigenic variability. So this creates a very complex immune landscape in which different PRRS viruses are circulating simultaneously. And you guys on the farms are trying to tackle PRRS that we're calling a single thing, but in reality, they're slightly different from one another. Because of this diversity that PRRS has, it's quite challenging to manage PRRS from an immunological standpoint. To add complexity to this, PRRS generates a very abundant non-neutralizing response, which is shown in the red line in this graph over here. And the neutralizing response shown in blue over here, it's short and it's not necessarily broad. Meaning that, yeah, the animals are going to have some sort of neutralizing response, but it's not going to be protective against other PRRS. So again, it creates a condition in which the circulation of these different lineages and sublineages of PRRS can thrive. Because of that, vaccines and live virus inoculation have been used to try to boost herd and replacement animal immunity, particularly targeted against specific PRRS viruses that may be circulating on a given farm. This is the rationale behind LVI. So the idea is that, well, if I'm gonna be immunizing my animals against this one particular PRRS, but there is some potential there that even though you are acting on the dams, on the gilts, on the sows, these benefits of vaccinating these animals might show up on the piglet population, on the grower population that is going to emerge from these animals that are being vaccinated now. So there is a potential that as an industry we could leverage dam exposure for piglet protection. So we're going to be acting, or we could act on the dam, expecting to see benefits on the piglet population. This is an ongoing study by Dr. Mark Wagner. And again, I'm going to mention him several times throughout my presentation because I want to recognize the key role that he had in developing this and making this study happen. So with that said, the objectives of our study were to evaluate how different levels of maternal exposure in dams influence infection dynamics in a nursery pig co-infection model, to assess whether LVI exposure during gestation influences the replicative fitness of co-infecting PRRS, and finally to determine if boosting gilt immunity in late gestation can reduce viral diversity and recombination in piglets. Essentially, we want to look, when we intervene on the dam, what happened on the piglets. Our hypothesis is that stronger maternal immunity shapes viral evolution and recombination patterns with greater effects on homologous viruses. So we expect that if we do expose the gilts, the dams, towards one particular virus, we're going to see a stronger effect on the piglets against that one particular virus. So this is just a drawing trying to illustrate a little bit of the study. I'm not going to spend too much time in here because I'm going to spend more time in the next slides talking about this. So for this study, we had 3 groups of gilts. And the way that I wrote over here, 3, 3, 1, in the first group we had 3 gilts, in the second group we had 3 gilts, in the third group we had a single gilt. These different gilts had different immune exposure protocols. And this is what this table on the bottom here shows. And I apologize for the folks in the back. I know that the table is a little bit smaller, but there's also space in front of the room. So if you want to see it better, please move forward. What this table shows, and I'm going to keep walking one side and another just because it's going to be easier to point this way. So this table has 7 rows. Each row is essentially an animal, a gilt. There's 3 groups over here, high exposure, low exposure, and control. Those are the levels of exposures of those gilts. And I'm going to enter into more details about what those mean. We had what we call low and high exposure plus control. You can see over here, pre-gestation, around 100 days of age, all of these animals, they got exposed to an LVI that was an L1C virus. So all of the animals got the same LVI prior to gestation. What's changed is that at 220 days of age, these animals were bled, and focused fluorescent neutralizing titers were assessed. And even though all animals were exposed the same, they don't all respond the same. Some of these animals had titers at 220 days of age, and we called those animals part of the high exposure group. Well, at this point they were high respondents, but they were assigned to this high exposure group because they were high respondents. And you can see on the low exposure group over here that all of the, and on the control, that all of these animals were negative at that point. At 48 days of gestation, we did killed vaccine on these animals on the high and the low exposure group using 2 different viruses, L1A and L1C. This happened on 48 days of gestation, and on 70 days of gestation, we inoculated them also with a killed vaccine of an L1C only virus. So these are the main distinctions up to now regarding the high and the low exposure group. At 90 days of gestation, we did 1 extra LVI on these animals, but only on the high exposure group. So again, to recapitulate this a little bit. Oh, I should be clicking my slides. Yeah, so majority of the exposure that we did over here was using an L1C virus, but not all. Please pay attention that there was, at 48 days of gestation, we did inoculate them with a killed vaccine that contained also a lineage 1A virus. So this figure on the right now tries to recapitulate and sum everything that I mentioned so far. We had 7 gilts. We are working with the piglets from these gilts. These gilts were separated into 3 different groups: low exposure, high exposure, and control. And the main distinction between these groups was that the low exposure, these gilts, they did not have focused fluorescent neutralizing titers at breeding, and they did not receive LVI at gestation. The high exposure group, they had titers at breeding, this over here and over here for this side of the room. And they did receive LVI at day 90 of gestation. So these are how these groups were formed. But we're not working with the dams, we're working with the piglets. Okay, so we used 9 piglets from each of these dams, each of these guilds, except for the control, which we used 5, but we end up not inoculating these animals. So what we did then was the following. We had 3 guilds on the low exposure group. Each of them contributed with 9 animals, 9 piglets to our study. We got 3 piglets from guild number 1, 3 piglets from guild number 2, 3 piglets from guild number 3, and we assigned these 9 piglets to one challenge group, that we challenged this group with a lineage 1A virus. And we did the same protocol with the rest of the piglets on this study. So this is what this figure shows. It shows that these different groups of piglets that we challenged, they came from different sows, but we tried to make the composition of the piglets from each sow to be equal in these groups. So we ended up working with 54 piglets from these guilds that were challenged with 2 viruses. The viruses, or the challenge groups that we created were, one of the groups was challenged with lineage 1A only, another one was challenged with lineage 1C, and another one with lineage 1A plus lineage 1C. It was a co-infection. We intentionally exposed them with both viruses simultaneously. And the same exposure protocol happened with piglets originating from high exposure guilds. We measured the focused fluorescent neutralizing titers of piglets at days 3 and 18 of age, so prior our challenge protocol, because we wanted to see if the exposure that we had done on their dams had any impact on them prior to our challenge. Oops. We bled these animals on day 0, 3, 7, 10, and 14, and then 14 was the endpoint for our study. That's when we euthanized all of the animals on our study. This top part of the figure illustrates that. We sent all of the samples that we collected from these animals for RT-PCR, for PRRS, and we did whole genome sequencing through Oxford Nanopore Technologies, sequencing only for day 7. That's only for day 7. That's when we had the highest viremia, the lowest CTs, so it was easier to obtain whole genome sequences from these animals at that particular time point. So going through results. So this figure, this slide over here shows the focused fluorescent neutralizing titers of piglets at 3 days of age, prior to our challenge. Again, at this point, we were just trying to evaluate if the exposure that we had done on the guilds, on their dams, were showing up in the piglets somehow. And yeah, we can see the piglets born to high exposure guilds, they had higher FFN titers at day 3 than piglets born to low exposure guilds or piglets born to control dams. The same thing happened when we looked at samples collected at 18 days of age from these piglets. The same conclusion. We can see that the titers from this group, although smaller, they're still higher than the other groups. Bless you. Another piece of results for us. We saw that piglets, after we challenged them, we saw that piglets born from both low and high exposure guilds got infected with PRRS. This figure on the right shows exactly that. It shows the individual piglet viremia trajectory over time for the different groups which they were assigned, high and low exposure, challenged with virus lineage 1A, lineage 1C, or both viruses. The lowest CT that we obtained was around 7 days post-infection in all of the groups. Again, that's why we use samples from that time point to obtain whole genome sequencing. And not all piglets had full follow-up. We had to do midpoint euthanasia for some of the animals, and 3 piglets, this was part of the protocol, but 3 piglets had early endpoints after day 7 because they were too bad. We decided to euthanize them before the end of the study. So a little bit more, okay, how can we contrast these viremia results from these animals, from these piglets originating from these different dam sow populations? So piglets born from high exposure guilds had higher CT values, lower viremia. So this is what this plot over here shows. It shows CT according to, from the piglets, according to if they were born from high exposure guilds or low exposure guilds. And you can tell that there is a distinction in the CTs, meaning that the CTs increase, you have lower viremia, depending or as a function of how much immunity or exposure the guilds that birthed these piglets were exposed to. This held true in all of the days. The plot on the left shows all of the data points for all days thrown together as if time did not matter. It does. So on the right, we do have the same plot, but now stratified according to the time after inoculation, DPI, days post inoculation. So this message that piglets born from high exposure gilts had higher CT values held true in all of the days of our study, as you can see on the second half of this slide. We also saw that piglets inoculated with an homologous virus, lineage 1C. Again, most of the exposure of the dams was lineage 1C. So we saw that piglets inoculated with this lineage 1C had a higher CT value, lower viremia. This is what this figure over here shows. There are 3 box plots over here, one of them for lineage 1A, another one from lineage 1C, another one for both. This sentence over here, I'm mostly comparing lineage 1C versus lineage 1A. And this is the p-value that I'm referring to. So lineage 1C had higher CT value, lower viremia, than lineage 1A. Viremia for lineage 1C and lineage 1A plus lineage 1C was comparable. Same story as the previous slide. Now I'm breaking these into days instead of just throwing, pretending that time didn't matter. Time does matter. This was true on most days. Not always, but in most days. So for example, over here, 3 days post-infection, there are no statistical differences between the viremia of this group over here, lineage 1A, versus lineage 1C. Although, maybe, well, of course, this is a power problem, right? Because, well, if we had enough power, we would have seen statistical differences. But there seems to be some sort of numerical distinction between the groups. At 7 days post-infection, there actually was a difference between the viremia of these groups. Same thing at 10 days post-infection, and so on and so forth. So piglets' viremia varies by the dam exposure and the viral challenge that these piglets are given. What do sequence reads tell us? Again, I mentioned that we did whole genome sequencing from these animals. So this plot over here, or this slide over here, shows the number of reads. So what I'm plotting over here are the number of reads per animal according to the group that the piglet was assigned to. So over here, the first graph, I'm talking about piglets born from low exposure gilts that received lineage 1A virus. So this is this blue color over here. If you look on this right plot over here, those are piglets born from high exposure gilts that also received lineage 1A. And in general, you can see that the bars seem to be a little bit smaller, lower, on piglets born from high exposure gilts on lineage 1A viruses. The same thing can be said on piglets exposed to lineage 1C virus, and maybe the same thing can be said on piglets exposed to lineage 1A plus lineage 1C virus. So yeah, we obtain fewer reads from piglets born to high exposure gilts, regardless of which virus they were challenged with. However, there's a lot of variability in the data, right? The way that I created this plot, again, I mentioned that we try to make the composition of these groups, the piglets on these groups, not come from a single sow. So the way that I plotted this, these animals over here, A1, A2, A3, B1, B2, B3, C1, C2, C3, they are siblings. They came from the same leader. So they came from the same sow. And then you can see that every group of 3 piglets over here, there seems to be some sort of dependency effect on them, right? And to me, it's very easy to see on the high exposure group. These animals on the first column over here, they seem to be, on average, animals originating from this leader over here, they seem to have more reads than other piglets on the same group. So yeah, it seems like we see some sort of signal over there, but it's not always true. It's not always true for everyone. So there seems to be a little bit of a complex effect here. So we think that there is a dam-specific effect. Piglets born to the same gilt tended to have similar number of reads, regardless of the lineage of the viral challenge. And that can be seen from piglets that receive lineage 1A, lineage 1C, or both of them, right? And on the others, you can also, if you squint your eyes a little bit, very statistical approach, it does help. It does help you to see that a little bit. Talking a little bit about recombination now, because I was just talking about read numbers, but what about recombination? So this table shows the number of reads and the number of recombinant reads that we got for each gene section for these low and high groups. We can only see recombination in animals that were challenged with both lineage 1A and lineage 1C because, well, that's the 2 viruses, distinct viruses, have to be infecting the same animal for recombination to occur. So it doesn't make sense for us to look for recombination in animals that are infected only with lineage 1A or lineage 1C. So the number of recombinant reads that we received we obtained from piglets born to the low exposure group was around 2,200, while from animals born to high exposure gutes was at around 1,000 reads. So we found more recombinant reads in animals born to the low exposure group. And that was true for all of the gene sections that we looked at. So this is what this proportion over here shows. The first column that I highlighted in red here is the number of recombinant reads, while the second column over here shows the proportion of recombinant reads for that particular gene segment. And this proportion over here on the first group of data here, the piglets born to low exposure gutes, is larger than the proportions for all gene segments on piglets born to the high exposure group. So it does seem like this exposure protocol that we did on the dams had even an effect on how likely recombination was on the piglets downstream. And that is interesting. Low exposure gutes produced piglets that allowed more viral replication. On the previous slide, I showed that the CT between these different groups was distinct, so that could perhaps help explain this finding, right? Recombination doesn't occur in a vacuum. The more virus you have within an animal, maybe you have higher odds of recombination to occur. So there's a lot of effects happening here at the same time. Another thing, piglets born to high exposure gutes cleared the heterologous, mostly heterologous, the lineage 1A virus, but not the homologous virus. Why? So this is what I'm showing on this figure on the bottom right of the screen over here. I have the proportion of reads according to the virus. So green is lineage 1C and blue is lineage 1A for each piglet that was born from low exposure gutes and from high exposure gutes. And you can only find lineage 1A viruses in the low exposure gute, not on the high exposure gute. Why? Could this be a higher fitness of lineage 1C virus? Yeah, maybe. But you would if that was only the reason, you expect to see this preponderance, this majority of lineage 1C viruses to be shown on both groups, and that's not what we saw. Could it be antibody-dependent enhancement? That's a mechanism that has been described for many diseases, even for PRRS, in which an imperfect immune response actually allows the multiplication of viruses because the immune response is imperfect. So maybe that helps us understand a little bit, but I'm just trying to find justifications. I actually don't have a very good justification for that. But it could be the case that because the immunity on the high exposure group is stronger, it allows, or it prevents the multiplication of one of the viruses. But why not for both viruses? I don't have a very good explanation for that. So we're not sure why we're seeing this. So going a little bit into the discussion. Piglets born to highly exposed gutes tend to have higher immunity levels. While exceptions exist, this is relevant information for practitioners. Again, if this is something that you're trying to do, you're trying to increase the odds that your piglets are going to have a good performance on their finisher phase. And if you think that pursuing a higher immunity level on them would help you, maybe this is one way of approaching this. Historically, talking a little bit about PRRS immunity and evolution, whenever people discuss about using vaccines, immunological interventions on these piglets, or in animals, you're going to see a lot of discussion focused on reducing clinical signs of animals, which is very important, improving performance, crucial, lowering viremia, extremely important, and increasing antibodies productions on these animals. All of those are great points, but I would like to make a case that those are not the only things that we should be looking at. I think that we need to assess vaccine impacts on viral evolution. This is a topic that we haven't discussed all that much, and it is possible that by doing these immunological interventions, which again, I'm not saying people should not use, that's not what I'm saying, but it is possible that they may have an unintended consequences to viral evolution. This is a paper from Nakarimpa Morchanavukun, he's sitting on the corner, in which he's shown that PRRS diversification was greater in vaccinated animals, regardless of which vaccine these animals received. We did this test with 2 vaccines, than in control animals. Essentially, animals that were vaccinated, virus diverged more than in animals that were not vaccinated. Again, I'm not saying that people should not vaccinate. What I am saying is that there might be unintended consequences of vaccination. This link over here takes to the paper. I strongly recommend people interested in this to read. It's a dense one, but it's a very interesting one. Repeated gilt immunization can lower pig viremia and reduce recombination risk. I spoke about that before already, so I'm not going to take longer on this. And increased immunity may accelerate viral change, as Paploski paper showed, but limit recombination, which our data seems to suggest. So the effects seem to be a little bit perhaps contradictory or at least complex. So exploring these dynamics can better guide PRRS control strategies. So when we were discussing about these animals, about this data, so what makes these animals, these gilts, respond so differently for the challenge or the immunization protocols that they received, but also to be able to pass that along to their piglets? That may be a genetic trait. And if that's the case, maybe selecting for that could have PRRS repercussions, or I think that this is something that we should be perhaps chatting a little bit. Further studies on these are needed, particularly on production impact and heritability of this trait. Again, I'd like to be very clear that we did not take our piglets. After inoculation, they only stayed with us for 14 days. So we did not assess production impacts, and that could be very, very interesting endpoints to also look at. So keep that in mind. This is a field-oriented research, right? Field practices can be complex. It's very hard for me to explain the different groups, the exposure done on the gilts, but we're actually working with the piglets. The piglets have different groups, and they went to different viruses groups. It's very complex. But at the same time, this is the type of thing that happen on the field. People do very complex things at the field. This very, as an epidemiologist, I like field research, but I see the value of more controlled experiments. The problem of translating very controlled experiments is that the field is not very controlled. It's a very dynamic situation. So in a sense, this variety of exposures, of effects that we're seeing, maybe this is closer to what people see in the field actually. So I'd like to thank Dr. Mark Wagner for this study. These animals came from a farm of his. So I appreciate the opportunity of working with this. Several limitations in our study, right? Limited number of animals. Again, we ended up working with 7 gilts only, 54 piglets. There's reasons for that, but I realized that those are very limited numbers. It would be great if we could have done this with more animals, but we couldn't. There are logistical limitations also. There was an absence of a true control group that got challenged. We had animals that we called them control. Those did not get infected because we needed them to be uninfected by the endpoint of our study. I would have loved to have a true control group, animals born from gilts that were not exposed at all, and just see what happens when these animals are challenged with lineage 1A, lineage 1C, and with both viruses. Oops. Generalizability. These gilts of ours, they received LVI and killed vaccines, which are very particular to each practice. Different people are going to use different LVIs, different people are going to look for different killed vaccines, if they use killed vaccines at all. So again, keep that in mind. This might not be fully transportable to every single situation, transferable to every single situation. I mentioned that already, but there's a complex immune landscape on our gilts. They were not exposed to a single virus. They ended up getting exposed to a killed vaccine that contained lineage 1A and lineage 1C at some point, so that might have introduced some noise. But at the same point, that maybe broadened their immune response. Maybe this also represents better what happens on the field. Piglets were not kept until market to assess production. I just mentioned that, right? It would have been great to keep these animals. I think there are very, very interesting endpoints or outcomes to look if that's the design that people want to look. I think that there's a great opportunity there to explore this. We only did whole genome sequencing from blood collected at 7 days post-infection, post-inoculation. Again, there's a reason for that. Those were the days in which we had lower CT values, and we can't really obtain, or the proportion of animals from which we obtain whole genome sequencing decreases when we tried doing that for other days post-infection. But it is possible that the viral population on other points of the infection could have been different. So my main take-home messages here today are that maternal PRRS to exposure may reduce viremia and increase antibody titers on the piglet population. And I think that this is important information. While immunization protocols may increase genetic mutation accrual, it may also impact recombination. Again, if this is something that you are worried about on your farm, keep that in mind. PRRS-related immunity management choices in the dam population likely have effect on piglets population. So you acting on the dam, on the gilts, on the sow population may bring benefits on the piglet population downstream. And perhaps the easiest way that I can put this out, although it's very simplistic, is going back to the CT values that we saw in these piglets, right? If you are one of those people that believe that CT values correlate with performance of these animals, this is one of the signals that was stronger in our data set, right? Animals or piglets born from highly exposure gilts had lower CT levels, lower viremia. Further research is needed to clarify maternal immunity role on PRRS dynamics. Again, we are putting our toes in the water. Is this the expression? We're just beginning to explore this problem. I think that our data suggests that there may be very interesting dynamics happening here. And I think that as an industry, there might be benefits of trying to tease those apart, pursue these effects in more detail. I'd like to acknowledge once more Kimberly van der Wal with major role in coordinating this study, the entire group that helped this study to happen, but again, Mark Wagner, this study would not have happened without him. I'd like to point, and I know that you're coming here, so I apologize, I'll be quick. I'd like to thank SHIC once more. SHIC is funded by a lot of you folks. I appreciate that a lot. A lot of the research that we can do is because of SHIC, because of these industry-oriented funding agencies. So yes, thank you so much. NSF and USDA that also helped provide funds for our study. Thank you so much.
Are there any quick questions?
I was impressed with the recombination findings of all your rates in the piglets, and I have two questions. If that happens in the field, how does that affect all the other lineage and variant classification published? Because those methods assume that there is no recombination.
Yeah, no, that's a great question. So recombination is a tricky one, right? Because it depends a lot on what viruses you have. Our ability to detect recombination is a function of how distinct these viruses are. When I mentioned, it's only worth looking at recombination in the group that is infected with both viruses. And I stand by that. I think that is true. It does not mean that recombination is not happening on the other groups. It's just that if it happens, it might not matter as much because the viruses that may be recombining are the same. So regarding recombination in the field, it really, really depends. If your viruses are too distinct, I think that the potential for something more distinct to show up, I think it's greater. So I don't have a very good broad answer. I think that how much does recombination matter in the field is going to be a function of context in each situation.
But how does that affect the lineage classification and all the other stuff if the recombination is happening?
So lineage classification is done based on R5 only, so you can have recombination happening outside of R5, so you can have recombination happening in R5. So the exact spot where recombination happens might have a certain impact on the classification or not. Keep that in mind. Classification of PRRS nowadays is done using only a gene, which represents 4% of the viral genome, and we have reasons to use that. But it's not an end-all, be-all. You do have a point. We may have recombination events that may not matter for classification because they're happening outside of R5. We may have recombination events that may matter for classification. Some of the variants that we are seeing currently in the US, they are a product of recombination at the R5 level. So yeah, it may happen.
We got one more question.
Yes, two very fast. You know the virulence level of your two different viruses to challenge, and the second, you search the virus in the lung and in the lymph nodes of the piglets?
Great questions. Both viruses, both lineage 1A and lineage 1C viruses were selected because they were perceived to be more virulent in the field. So those were viruses that people used to call lineage 1A 174. People may be familiar with that. And the lineage 1C virus is a lineage 1C.5. So it's one of those that emerged maybe 4 years ago, 5 years ago. Both of them were chosen because they are perceived to be more virulent. I don't have data on virulence, but that's the reason why we chose them. And the second question of yours was associated.
If you search the virus
Yes.
in the lung.
Perfect. So we did collect tissue from these animals to look for viruses in these tissues, and the information that I got from the lab is that they could not find virus in these tissues, which surprised me a little bit. So we're trying to discuss this a little bit more. But yeah, that's the information that I have.
All right, let's thank Igor.